All right. So, ladies and gentlemen, hi. Hello. I know there was a test or there will be a test of some other class, but I will ask you just try your best. Focus here. Okay. Um, yep. Now, also today for us, we're going to be talking about this topic. We're going to stop at gametogenesis, finish gametogenesis, and then probably in the last 15 minutes of class, I'm going to give you information about the exam. Okay, so when we're talking about in this handout, when we talk about gametogenesis, we are talking about sex cells being made. It is a term you will use for both male and female. It will be a term that you use for both male and female. Okay. So when we're talking about gametogenesis, we are going to be talking about it includes spermatogenesis and oogenesis. Spermatogenesis and oogenesis. Okay. So process of making a sperm, process of making eggs or ovum. Now, we will need to look back a little bit on the process of meiosis last year. You guys all learned meiosis last year. That is fine. So what I'm going to do is I'm going to refer to certain portion of that of the grade 11 textbook so that we can kind of remember what happens. Okay, we can remember what happens. And at the same time, now you don't need to quite exactly remember in this course, right? For this course, you don't need to remember like, oh yeah, in meiosis, in prophase one, what is happening? You don't prophase anaphase two, what is happening? You don't need to know exact details of what's going on. Uh, we're going to refer to those stages. Okay, we're going to refer to those stages, but it's just I'm showing this one so you kind of like, oh yeah, so this is where it is. Hopefully, it'll make things easier to understand. Okay, so let me first. Now, when we're talking about meiosis, let's do a quick review. What do you start with? What do you start with? Or how many first? How many of what do you start with? This is. You start off with what, guys? Sorry, say again. 23 chromosomes? 46. Now, you start off with how many of these? Now, we say 46 chromosomes, or another way we want to say it would be 23 pairs. We like to say 23 pairs because it kind of reminds us, oh yeah, half of that is from mom, half of that is from dad, and you can have homologous, okay, chromosomes. So in this case, 23 pairs of 46 chromosomes. Now, if you have 46 chromosomes of 23 pairs, what end do you have? There's a ploid. What ploid are you? Anyone? Okay. Anyone remembers? Yes. What? Diploid, 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 diploid. You have a diploid 2n. Okay. How many of so for meiosis you have 46 chromosomes, 23 pairs, diploid germ cells. Okay. Germ cells is just a very generic term to say what this is. Keep in mind, when we're talking about spermatogenesis and oogenesis, you will learn a different name. But for now, just germ cells. At the end of meiosis, one becomes what? One becomes, don't remember. How many in meiosis? Like how many uh how many phases does it have? No one remembers. Oh boy. Okay. For time's sake, 4. Okay. You just a reminder, after meiosis 1, 1 becomes 2. Okay? And then you have meiosis 2. Okay? PMAT1, P1 M1 A1 T1. You have two cells, and then the same thing again, but P2 M2 A2 T2. You have 4. Okay. You have 4 of what? Let's start with the chromosome stuff. Yes. Okay. So let's talk about chromosomes. I'll come back to you. How many chromosomes in each of these things? Yes. Just 23. Good. Right underneath here. So you have 23 chromosomes. And at this point, what ploid are you? What ploid are you? What is half of diploid? What? It's not my. The reason why I say half of diploid is because it's haploid. I did not make up the system of naming these things. Okay? Haploid. And you don't have germ cells, you have gametes. Okay? Sex cells. So that's the process of meiosis. Good thing that we go back over it. Okay? I'm just looking for the diagram so that hopefully we can remember 130s. So thankfully, you don't need to remember every single stage, what happened at what. Okay? However, I'm just going to refer to each of those. Okay? Yes. Oh, good. Who was your teacher last year? Ooh. Okay. Okay, so some teacher in grade 11 did teach that. I know that others might have, and I know others didn't. So yes, when during meiosis, okay, during meiosis is 1 to 4, okay, 1 to 4. But when we're talking about spermatogenesis and oogenesis, the polar body stuff, That does happen. So yes, it is different for guys from girls, and we're going to talk about it. Okay, but I'm glad that she brought that up. Any of you guys had miscaps last year for grade 11? No. Okay. So let's go here. We're going to start off with the guys first. Okay, we're going to start off with the guys first. Okay. Oh yeah, the diagrams. So meiosis, one, remember, you start off, this is meiosis one. You start off here, these two, you start off with two daughter cells, but they're not genetically identical to each other, okay? So meiosis one, and then afterwards, meiosis two, okay? For haploid gametes. Okay. Take a note that for each one of these, you basically have four, and then later on it separates. Okay. So let's talk about the guys one. Okay. So spermatogenesis. Spermatogenesis makes spermatozoa. Spermatozoa, that's a technical name for sperm. Ova is the technical term for eggs. In that is Latin. So ova is plural. Ovum is singular. Okay. Ova is plural. Ovum is singular. Are we good? So let's look now, because this photocopy is black and white, so it doesn't show the color that well. So I'm gonna refer to the electronic original here. You can access this in the Google Drive folder, okay, which have the handouts. It's there, okay. So this here is a cross section. So if you take a testes and you cut it like this, you have a cross section of the testes, and you have the epididymis right around here. Now, inside the testes, there are a lot of coils of tubes of these seminiferous tubules. And inside these tubules, if you cut the cross section, okay. Now, in the middle, that's called the lumen. Okay, we use the term lumen for pretty much every time we talk about a tube, blood vessel, digestive tract, the hole in the middle is a lumen. Okay, so when we talk about lumen down here, that's what it is. If you look at this section here, now you have these cells here. These cells are starting to call the spermatogonium. And you have these cells here, these little blurb here, they are called the Sertoli cells. Sertoli cells are supposedly the supporting cells. They play a supporting role in terms of the spermatogenesis, but without it, it won't happen. The spermatogonium are the ones that later on go through mitosis and meiosis, become sperm itself. Okay, how are we doing? Okay, so if you also look at the handout, what you will see is this. I'm going to refer to the handout. I'm going to start writing on things too. So if you look, so the diploid spermatogonium, which is these cells here, they are the germ cells. They're the germ cells. They are diploid. They will go through mitosis, and then it becomes primary spermatocytes. Okay, primary spermatocytes, which is 2N diploid. So primary spermatocyte would be genetically identical to the spermatogonium. It's mitosis. You're making copies. So it's still 2N. The diploid primary spermatocytes go through meiosis 1 to make 2 haploid secondary spermatocytes. So primary spermatocytes go through meiosis 1. So instead of diploid, it now become haploid, 1N, and there's 2 of them now. Okay, so secondary spermatocyte is a haploid cells. Secondary spermatocyte go through meiosis 2 to produce 4 haploid spermatids. So again, each of these becomes a spermatids. Okay, meiosis 2. So spermatids receive nourishments from Sertoli cell and differentiate into spermatozoa, growth flagella, acrosome, etc. We're going to talk about what these things are a little bit later. Flagella and acrosome. You should know what flagella is, okay? So, spermatogonium go through mitosis, makes primary spermatocytes. Primary spermatocytes go through meiosis 1, secondary. Secondary spermatocytes go through meiosis 2. You have spermatids. Spermatids with the nourishment from Sertoli cells make spermatozoa. Okay? Are we good so far? Now, after that, once it's done, the spermatozoas will detach from the Sertoli cells, move through the lumen of the seminiferous tubules to the epididymis. So we'll go through these tubules and get to epididymis. Now, let's see how much you remember. What is the role of epididymis? I know we did look at that a number of lessons back. What is the role of epididymis? If you made something, but you're not using it, what are you doing with it? It's just sitting right there. You made it, you're not using it right away, it's just sitting right there. You are, yes. Oh, is that hand up? No. You want to take a guess? Storing it, exactly. And the epididymis, one of the function is storage. Okay, storage. And the other function of epididymis, during epididymis, sperm will mature there and gain something. It sounds like a way to talk about cell phones. Yes. Motility, thank you. Okay, it will gain motility. It can move. Okay, mobile phone, motility. Okay. Actually, in period 1, we got into a whole discussion. What's the difference between mobility and motility? Apparently, motility is about cells and body parts. So that makes sense now. That's why we use motility. Okay. So these spermatozoas go to the epididymis. This is where they will gain the motility. After all, yes, they have the flagella, but they have to be able to do a number of things before they can swim a relatively long distance. Okay. Are we good so far? Outside of the tubules, there are blood capillaries and interstitial lytic cells. Now, it's not shown in these pictures. Okay. Now, lytic cells, the role of it is to make testosterone. Okay. And we did talk about testosterone, the secondary sexual characteristics last lesson. Okay. On Wednesday. Are we good? Now, we are going to refer, now what, I didn't print it out. Please write it down here, okay? You will need to look in your textbook, page 501, figure 5 of your textbook, okay? Page 501 of the textbook, figure 5. It talks about the hormonal control and regulation of spermatogenesis, okay? So we're talking about the grade 12 textbook, not the grade 11 textbook. Okay. Now, hormonal control or regulation of spermatogenesis. Before we get there, okay, while you're copying that down. Now, there is a reason why we talk about the menstruation cycle first. Okay, there is a reason for that. Okay, A, in some ways it is more complicated, and 2, now let's see, what are the 4 hormones? What are the 4 hormones that we have learned from the menstruation cycle? Yes. Progesterone. Good. FSH, LH, estrogen, progesterone. Can you guys remember who make FSH? Or I shouldn't say, what makes FSH? Which part of the body make FSH? What? No, say it. Try to say it. Yes, good. Pituitary gland makes FSH. LH. What makes the LH? Also pituitary gland. So pituitary gland makes FSH and LH. What makes estrogen? 2 things make estrogen. And one of them, after a certain event, become the other. Anyone remembers? Now think about menstruation cycle. There's a very key event. Before that key event, this thing had a name. After that key event, it becomes a different name. What is that key event? Menstruation cycle. How much you guys remember? It's helping you to review right now. Okay? So if you actively participate, it's a way to review. Menstruation cycle. What is a key event? What's the point of menstruation? What? Someone say something? What? I can't tell. You guys are all wearing masks. Ovulation. Good. So key event in menstruation cycle is ovulation. So before ovulation, this thing has a different name. What is the main? Who's the main key player of ovulation? Guys, ladies, gents? What? Okay, what does FSH stand for? Follicle stimulating hormone. So before ovulation is the developing follicle that makes estrogen. Okay? After ovulation, the ovum exit, you are left with this thing. What is this other thing called? It has a Latin name. Yes. Good. Corpus luteum. Corpus luteum. Okay? So, developing follicle, corpus luteum makes estrogen. Who makes progesterone now? is made by this thing with a two-word Latin name, which we heard just now. Yes, corpus luteum. Thank you. Okay. Definitely need to review that section, guys. Okay, so FSH, LH, estrogen, progesterone, and pituitary gland, uh, developing follicle, and corpus luteum, corpus luteum. Now, the last two, they're exclusively only in female, estrogen and progesterone, so to speak, okay, for our course. So in this case, they will be considered female sexual hormones, okay? Now, FSH and LH, they are made by the pituitary gland. Guys have pituitary gland too, thankfully. So in this case, guys would also make FSH and LH. We just don't make estrogen and progesterones. Are we good? Okay. Now, the textbook. It's to build up to help you guys remember all these things, okay? Oh yeah, I actually took a picture of this earlier. Okay, good. So, textbook here. I just took a picture of this. I'll add the picture of this onto the handout folder. You can access that later on the Google Drive if you want. But basically here, so you have the hypothalamus, which is part of the brain. Okay, it's kind of sitting right in the middle of it. Connect to it is the pituitary gland, but the pituitary gland separates into two parts. The front part is anterior pituitary. The back part is posterior pituitary gland. We're not going to care about that right now. The main one is anterior pituitary gland. The hypothalamus releases a hormone called gonadotrophin releasing hormone. Gonads is just a very neutral way of saying sexual organs and parts. So in this case, hypothalamus in both guys and girls are releasing GnRH. GnRH in guys will go to the anterior pituitary gland, and anterior pituitary gland will release FSH and LH. Just right now, this is the same for guys as for girls so far. Now for guys, FSH, when it gets to the testes, okay, let me just zoom in a little bit more. By the way, you have red arrows here and green arrows with a plus sign. Green arrows with a plus sign means stimulates, okay, the release or stimulates. Red arrow with minus sign, that's inhibits. So FSH will go to the Sertoli cells, okay, Sertoli cells that we saw earlier. The Sertoli cells will start producing a protein, and that protein is needed basically to stimulate the process of spermatogenesis, okay? This is the reason why I said before, Sertoli cell look like it's playing a supportive role, but without it, it's not going to happen, okay? So the FSH send a signal to Sertoli cell basically to stimulate, kickstart the whole process of spermatogenesis. The LH in guys will go to the Leydig cells. Now, you didn't see the Leydig cells. It's not inside the seminiferous tubules. It's outside of it. Leydig cell will make testosterone in the testes, and that testes will be released into the bloodstream. Now, we talk about what testosterone does, okay? A lot of the secondary sexual characteristics that we talk about on Wednesday, including sperm production. Now, there is a feedback loop, okay? in all biological systems, there are always a feedback loop. The Sertoli cells will make a hormone called inhibin. Inhibin will inhibits the release of FSH in the anterior pituitary gland. Okay, because you don't really need to make too much FSH. If there's too much is made, you don't want that. So you want to just limit that, inhibits that. If there is too much testosterone, yes, there is a thing, then it will have a negative feedback. It will stimulate Sertoli cell to make more of this inhibin, then in this case, really slowing down the whole process. Also, the testosterone will inhibits the release of LH from the anterior pituitary gland. Last but not least, the testosterone will also go to the hypothalamus to inhibit the GnRH being released. Okay? So make sure you know this hormonal regulation system for spermatogenesis in guys. Any questions so far? Now, one thing. Yes, it is hormonal control. It is hormonal control. Yes, there's stimulation and feedback and inhibitions, but spermatogenesis, unlike oogenesis, it's an ongoing process. It's an ongoing process. Okay. So it doesn't stop and wait, unlike oogenesis, which is like once a month, a few of the follicles get developed. Good. Okay. That's spermatogenesis. Any questions? Okay. Make sure time. So a few things. The structure of sperm. Okay. The structure of sperm. You have a head, you have a mid-piece, you have a tail. Sperm cell is probably the most recognizable human cell because of the tail. Okay. Now, the head contains a number of things. The haploid nucleus contains the dad's DNA. You have the acrosome cap. This is important. Acrosome cap contains hydrolytic enzymes which help sperm to penetrate the jelly coat of an egg. You're going to learn about the structure of the ovum of the egg. There are a number of coating outside of the egg. Okay, so in order for the sperm to go through to fertilize it, it has to penetrate that. You need the acrosome enzyme. You have the centrioles needed for the zygote to finish, to divide. The mid-piece is mainly mitochondria. Keep in mind, this sperm needs to swim a relatively far distance. You need that mitochondria to make ATP. Last but not least, the tail is just to facilitate movement, just like all flagellas. Are we good? Okay, oogenesis. Okay, so oogenesis started off as a fetus. Okay, started off as a fetus, but once the baby girl is born, she would have all the eggs that she would ever have in her whole entire lifetime. Okay, and the cells that we're talking about is oogonia, and that's plural. In singular is oogonium. Oogonium. So these oogonia will go through mitosis to build up. Okay, that's before birth. Now, these oogonia will turn into basically it's just grow bigger. There's not much different from it aside from size and content. Okay, the genetic information is identical. So by the time it's born, these primary oocytes, it will stop in prophase 1, pretty much right in the beginning of meiosis. Okay. So when the girl is born, they have all the primary oocyte. They will all stop in prophase 1. Nothing happens during childhood. Okay, ovaries are inactive, so to speak. Starting puberty. Okay, so during hormonal, for each menstrual cycle, this is the part where earlier Mia, you were saying about polar bodies. This is the part that happens in girls. So in this case, now every once a month, a few oocytes will go through meiosis 1. Okay, only few. Okay, now, however, the two resulting haploid cells, the two resulting haploid cells are not equal in size because the cytoplasm will mostly go to one of them and not the other. Okay, most of the cytoplasm will only go to one of them. The one that didn't really receive any cytoplasm, they become polar bodies and they just die off. Okay, yes, it has the chromosomes, but it's not going to be used. Die off. And that secondary oocytes will continue and it will go through prophase 2. That is still inside the ovaries. So secondary oocytes will start and finish prophase 2, but it will stop at metaphase 2. It will stop at metaphase 2. So by the time ovulation happens, that ovum is released. That ovum actually have not finished meiosis yet. It stopped at meiosis 2. If there is no fertilization, then menstruation will happen. So when menstruation happens, basically the ovum that comes out only goes to metaphase 2. Didn't even finish. Now, if there is fertilization, then this secondary oocytes will actually complete the rest of the meiosis. Now you'll have a second polar body. It will receive the. It wouldn't receive any of the DNA from the sperm, but then the rest of that, it becomes second polar body, and then you end up having the zygote will be fertilized and then becomes the germ cell will become fertilized and becomes zygote. So by the end of, if oogenesis go through the whole entire thing and ends with fertilization, you end up with a zygote. This zygote will be a diploid cell, not a haploid. The reason why it's not a haploid because it had received that the other 23 chromosomes from the sperm. Okay. Any questions? No. Okay. So Now we're going to watch a video, and this video would actually be a pretty good, would actually be pretty good in terms of leading us to the pregnancy section. Okay, so now when I watched the video, I watched it without sound and just subtitles, so I totally did not know how the audio sounds like. So if you're if you find it funny, that's why. It sounds almost like the Amazing Race. It should be a couple of millions. I don't know why they use thousands. So actually, if you go back a few slides earlier, you see like a whole bunch of sperm cells already at the ovum. But the thing is that that doesn't matter. If you haven't gone through it yet, it doesn't count. Okay. Okay. So basically, the oops, what did I do? So basically, the acrosome member enzymes and the acrosome was, remember, it's to break down the jelly coat of the ovum. That's what it was doing. Okay. So that's when the whole second part of meiosis two, my, metaphase two, anaphase two, and telophase two happens. Okay, we're going to stop here. Okay, we're going to stop here. Uh, and we're going to kind of finish this next week when we're talking about pregnancy and all that stuff, fertilization and pregnancy and all that stuff. Questions? Okay, the exam. So I I think yesterday on yesterday I received this email. Apparently, either you and your parents would have received an email about talking about exam and stuff like that. This email, of course, tell you the fact that yes, uh, the exam is worth 30% of your final mark. Okay. Uh, and we do know that this is your first time writing your exam. So in this case, uh, we have taken that in mind when we're designing the exam. Okay. There are a couple of things, tips, and all that. Please make sure you look through it and use that. Okay. They are very important. Now, in terms of the information of the exam itself for us, um, this is the first page, basically the first page of the cover sheet of the exam. So, can you look over this first? You can read. So, It is a paper and pencil exam. By the way, for those of you who are, I am recording, this is being recorded, so you can always watch this section later on. Okay. Now, one thing, any questions so far about the exam? One thing, we are it's paper and pencil. We're using bubble sheets. So for some of you, for some of your other science teachers, they have been using like, is it like a bubble sheet for multiple choice by Zip Grade, that kind of thing? Have you guys seen that? It's like that. So make sure you bring a pencil and eraser. You will need to shade in the entire bubble, okay? Because I'm just using an app and taking a picture of it to mark it, okay? So you make sure you shade in the whole thing. And if you need to make a correction, use a eraser, clean it properly, entirely well before you shade in the otherwise. Otherwise, when I take a picture, the app will think, oh, this person gave me 2 answers. That's wrong, okay? So make sure you have pencil and eraser. You will not use calculators. There will be no calculators for the biology exams, so don't bring one. In fact, the cover sheet will also say calculators are not allowed for the exam, and the teacher knows that. Are we good? Now, part C and D. Let's look at that first. So part C, this is, you have to do all these questions, okay? Do all these, you have to do all the short answers. So it's evenly split between Unit 1 and 2, Unit 3, and Unit 4. Now, the type of questions that I will be asking a lot of times is like, distinguish between these two kind of things. Explain how. Describe function of the following, and I'll give you things. You tell me what it does. Describe components of a process. Okay? So those are the typical questions. Part D, you have a choice. There are 3 questions. One question will be on Unit 1 and 2. One question, Unit 3. One question on Unit 4. Again, 5 mark, 5 mark, 5 mark. The choice is to give you a chance in some ways to like, oh, I think I'm out of these. You can take a look at the questions first, and then you see like which one you know you will get the better answer of. Or, hey, I know Unit 3 really well. I will go for that one. Okay? You have a choice. The typical question for Part D, I will give you a data or some scenario. You use the knowledge that you have to answer the questions. Okay? A little bit application, so to speak. Outline the process or roles of things in process. Okay? Important concepts for C and D. Enzymes, enzymes, enzymes. Make sure you know enzymes. DNA replication, transcription, translation, mutation. Make sure you understand those things, how it works. And of course, these 3 processes. Okay? Make sure you know them for C and D. Any questions so far? Now, the rest of it, A, B, as well as E, they will cover all the units. They will cover contents of all the units, unit 1 to 4. However, we know that this is your first exam. Typical exam would normally be, if it covers the whole year, it will be like, 25, 25, 25, 25%. Okay? It will be even split or pretty close to it. I'm aware that this is your first time writing culminating cumulative exams. So the marks are heavier, leaning to Unit 4 and Unit 3. Things that are either was doing or recently doing. Okay. So these numbers mean the following. For Part A, 2 marks on Unit 1, 2 marks on Unit 2, 2 marks on Unit 3, 4 marks on Unit 4. Okay. For Part B, the matching, 3 marks Unit 1, 5 marks Unit 2, 2 marks Unit 3, 5 marks on Unit 4. Okay. I was tired when I was doing this, counting this part. So there is an uncertainty level of plus or minus 1. Okay. But again, you can see it is leaning heavy to Unit 4 and Unit 3 on the multiple choice section. Okay. Because if I evenly split it, it will be a lot harder. Any questions? What I will suggest you start doing, that's why the lab report is due today, so you can start looking through your notes. Looking through your notes. Now you know that it's heavily lean on Unit 4 and Unit 3. Look through your Unit 4 and Unit 3 things. Look through your Unit 4, Unit 3 things. Start making study notes. Start making study notes. I will talk a little bit about making study notes next week too. I will finish off fertilization and pregnancy first, and then we end that, and then we will be doing review. But even now, you should start reading through things. Okay? Look through all those unit planners that you can still access from Brightspace. Okay, remember, if you go to Brightspace, the announcement, this is where you have all the unit planner links. So click at them, look through them, look through your notes. Okay. Any questions? Okay. Study your past tests. Okay. Especially the written stuff. Study the past tests. The review questions, textbook review questions from the past I assigned you guys to do them. Okay. I will next week. I'll go through things with you. I will try to give you guys more practice stuff to do. Okay. And talk about how to make study notes, because I'm pretty sure some of you just reading through it and it's not the most effective. Okay. Questions?